Hi, now we want to review our Genetics 101 for molecular principles in BME. So let's think about the genetics and inheritance. So Greg Mendel's discovery of these principles of genetics was revolutionary. Even before hundreds of years ago, knowing about these molecular principles of the DNA and RNA. So that is a fascinating. The importance of the discovery of the Greg Mendel is not about the experiment itself and his interpretation, but the power of by bringing a theory of how this inheritance works and predict the future. That's the power. That's what we acknowledge him as the father of genetics. So the genetics is a study of heredity and hereditary variations. And we talk about gene a lot. And what is gene? Gene is a locatable region of this genomic sequence first. And so a unit of inheritance as a gene, which is a locatable one, in somewhere in the chromosome. And so these genes are located on the chromosome, and that's within the nucleus. And this double-stranded DNA is packaged with a histone protein to form a material called chromatin. And let me go through once again. So DNA packaging in the nucleus of a cell. So people say who work with DNA call DNA as very sticky. The reason is, remember that DNA is composed of this nucleotide with a pentose sugar molecule and phosphate and DNA base. This phosphate has PO. There's a lot of negative charge, so DNA is negatively charged, and this makes DNA to wrapping around histone. Here, this is an histone to form a nucleosome. And you can see here, nucleosome. And nucleosomes are further packed together to form a 30 nanometer chromatin fiber, and additional levels of packaging result in highly condensed chromosome structure. So think about this real dimension of this whole process. You know, in the past, we only use an optical microscope to see micron level, which is this chromosome. When it's metaphase, it looks like this. It's 1.4 micron, so people, humans, can see the shape. But DNA is so tiny. Can you see this? This diameter is about 2 nanometer, and that's DNA double helix actual size. And that's wrapping around a base. It's a histone protein, and this package into nucleosome about 30 nanometers, still far small. And even this will become a scaffold onto chromosome scaffold to form about 300 nanometer, which is about kind of a good microscope can barely see. And it condensed with this form, and we call as a coiled coil structure. This is just for reference, but to give you an idea that DNA double helix 2 nanometer, at the simplest level, this chromatin is double-stranded helical structure of DNA. And this is wrapping around histone proteins, which is composed of 8 histone molecules and about 1.6 times rotation on average. And with this, it forms a chromatosome and nucleosome structure forming about 30 nanometer. And that forms loops averaging about 300 nanometer. And with this, about 300 nanometer fibers and fold to this. And this coiling results in to this final chromosomal structure, which is visible with our microscope. So in the past, so we have some genetic problem at the level of chromosome, people can actually find out. A good example is Down syndrome you can recognize the people which is affected by this, and people look at their cellular nucleus and those chromosomes, and people realize this. There's a 21 chromosome, there's a not only 2 but 3, so we call it a trisomy. So this observation of human chromosome is called human karyotype. So with this, we can start to look at some of the genetic heritable diseases. So these chromosomes can be isolated from a cell, and we can display it in a karyotype. So it's very simple. You just look at them and then sort them based on the length, 1, 2, 3, 4, in a shorter order. And then, so you can find pairs of this chromosome. One's typically from father, the other from mother. Okay, so this is as a 20 totally this pair. And this genetic makeup of an individual we call genotype. But the observable traits in an individual is called phenotype. So genotype versus phenotype is a very important discussion in genetics. And there are still, we do not understand. Some people argue that every disease will be a genetic disease, which has its own foundation too. So this karyotype has 23 pairs of homologous chromosomes, and the nuclei of all these somatic cells. Somatic cell means in our body part in comparison with the germline cell, which is coming from our sex cells. Okay, so nuclei of all somatic cells will contain, you can see this 22 identical, while the sex chromosome in this case is a male, so it's XY. And you know that for female, it's an XX. So these somatic cells contain 22 identical pairs of chromosomes plus a pair of sex chromosomes. And this chromosomal level aberrations, which can be observable with a microscope, we call it as a karyotype analysis. And for example, this number 12 chromosome, so one from father, one from mother, and these is a pair as a homologous pairs of chromosome, and the genes of one chromosome is called allele. So there are 2 alleles for the character, and the location of this on the chromosome of certain entity, certain genetic unit is called the gene, and that's a gene locus for that character.